Gene transfer to eukaryote has got many applications. It is used for creating genetically modified organisms. The methods used for transfer of DNA into the cell in prokaryotes is similar to that of eukaryotes, but it is different technically and procedure wise. Two major reasons for difference is like the eukaryotes is having a separate compartment as the nucleus, so the gene transfer method should be able to insert the DNA or deliver the DNA into the nucleus. So it has to pass through the plasma membrane to reach the cytoplasm. After that, it has to pass the nuclear membrane to get inside the nucleus. Second reason is like the compared to bacterial cells, the eukaryotic cells are very sensitive. If you are using a harsh technique or harsh chemical, the cell will die. So our technique should be precise as well as very gentle. Then only we will be able to attain the gene transfer to eukaryotes. So just like in prokaryotes, we can use the physical methods, we can use chemical methods, and we can use virus or bacteria to transfer DNA into the cell. So the DNA can be either inserted directly into the cell using physical methods, or most of the other methods resulting in DNA delivery into the cytoplasm, and there are cytoplasmic mechanisms which can actually transfer the DNA into the nucleus. So the transfection is actually the term we given to the introduction of foreign genetic material into a eukaryotic cell. It's a eukaryotic specific term, transfection. So any methods, the physical or chemical method we are used to insert the DNA into a eukaryotic cell, it's called a transfection. So in our discussion, first we will discuss about the gene transfer methods for animal cells. In the following video, I'll be talking about gene transfer to plant cells. And remember that most of the techniques are similar, but the way we are using the technique is different. And this both for animals and plant cells, we also have some specific methods which is only applicable for these cells. So gene transfer methods can be divided into transfection methods and biological methods. In the biological methods, we can use the transfection using virus, or we can use bacterfection using the help of bacteria. In the transfection, we have the chemical methods or physical methods. These are called direct gene transfer methods. So the chemical transfection is divided into three different methods. The chemical transfection using calcium phosphate method, transfection with the polyplexes, and liposomes and lipoplexes mediated gene transfer. So I'll be talking in detail about each one of this. The first method is calcium phosphate precipitation. This method is actually similar to the calcium chloride method or chemical method we are used for bacteria. But here we are using calcium phosphate instead of calcium chloride. So the methodology is like DNA precipitation, DNA molecules after precipitation is dissolved in a phosphate buffer. The phosphate buffer is having phosphate. After that, we will be adding the calcium chloride into the medium. When we are adding the calcium chloride, the calcium from the calcium chloride will be binding to the phosphate in the phosphate buffer to form the calcium phosphate. This calcium phosphate actually co-precipitate the DNA molecule into the bacterial surface, or not the cell surface of the eukaryotic or animal cells. Once it is in the surface of the cell will take up the DNA, which is in the precipitated form, using the phagocytosis. So as we know, the animal cells are able to phagocytosis the DNA, so it will be phagocytosis they precipitate to the cell. So by phagocytosis the DNA will get inside the cell. So this method is actually applicable for cells which is actually growing in monolayers, but if the cells are growing in clumps, this method is not that much applicable. We can actually use the different buffering system to increase the efficiency of transformation, and we can actually reach a transformation efficiency of 20%, which is actually really high. It is applicable to most mammalian cells on large scale. Second method is using polyplexes. Polyplexes mediated gene transfer is an efficient method of gene transfer. So what is polyplexes? Polyplexes are polycationic components. So the polycationic means it will have multiple positive charges. As we know, the DNA is polyanionic. It has so many negative ions in the form of phosphate. So these polyplexes are actually polycationic components. Our DNA is polyanionic. So when we are mixing the polyplexes with the DNA, it will form stable complexes because one is positively charged and another is negatively charged. One such example for polyplexes is the diethyl aminoethyl dextran. It's a water-soluble polycationic polyplexes. So once we mix the diethyl aminoethyl dextran, that is DAED, to the DNA, they form a stable complex, and it will be in contact with the cell, and it will take up the cell will take up this complex by using endocytosis. In the previous method, the cell is taking the phosphate precipitate, the calcium phosphate DNA precipitate using phagocytosis, but here they are using endocytosis. So this is the structure of dextran, DAE dextran, DEAE dextran. So this one has the multiple positive charges because of this amino group. So it will be, here is the representation. Plasmid is negatively charged. The polydextran is a polyplexus having positive charge. Once we mix it, we will get a like a complex. The complex will have a negative positive charge. So the cells will take up it using endocytosis. The disadvantage with this method is like the stable transformation is very difficult or rare, but it is very suitable for coarse cell lines. We have learned the coarse cell lines in SP40 vectors. We can combine this polyplexes along with the osmotic shock. Instead of using heat shock, here we can use, say, an osmotic shock. So in the osmotic shock, the cells will be transformed from a hypertonic solution into a hypotonic solution. So the osmotic shock actually increase the transformation frequency, or exposure to the cholequinine inhibitors will actually increase the transformation frequency by changing the acidification of the liposomes. Because after the endocytosis, the endocytic vesicles is actually binding with the lysosome. So if it actually binds to the lysosome, it will acidification occurs, and the enzymes will cleave the DNA. To prevent that, we can use the chloroquine, which is actually used to prevent the acidification of the vesicles. Polyamines are related to the polyplexes, but in the case of polyamines, it is having multiple amide groups. So it also have the amide groups also having the positive charge. Just like the polyplexes, it can actually bind to the DNA and form a net positive charge, and cells will take up this polyamines like endocytosis. The mechanism is similar, but here we have polyamines instead of polyplexes. The next method is liposuction. So as the method tell, the name cell is like infection with the lipids. So in this liposuction, DNA is delivered into the cell via liposomes or small vesicles we prepare from the lipids. So the lipoplexes, we can use lipoplexes that can be cationic or neutral in charge. Once we mix these lipids, just like bipolar lipids, which is having a positive charge or the charged components and a hydrophobic region, they will form micelles or vesicles with the DNA in a random manner, in a like spontaneous manner in the air. So these vesicles can actually interact with the plasma membrane and deliver the DNA. We can also induce them fusogenic lipids, which will actually fuse to the DNA membrane inside the DNA. So we have two different mechanisms. Either the DNA, if we mix the bipolar lipids with the DNA, it can actually form the vesicle. So inside the vesicles we have the DNA, and the polyplexes will be having a net positive charge, and our plasma membrane is negatively charged. So they interact each other, and it will take up the DNA by endocytosis, and inside the cell, it will be released into the cytoplasm, and eventually it will go to the nucleus. That's the one mechanism. The vesicles is taken up by the cell by endocytosis. Or we can incorporate some fusogenic lipids into the vesicle. If the vesicle is having fusogenic lipids, they won't get endocytosed. Instead, they will fuse together. So once the membranes are fused, the DNA will be delivered into the cytoplasm. So these two mechanisms, either using endocytosis or by using the fusion, lipoplexes or liposomes can deliver the DNA. The advantage of this method is like it has very low toxicity, simple to perform. It is applicable to almost all type of cells. It facilitates transient as well as stable transformation. Efficiency is high, and it can be used for transforming animal cell lines. Okay. Other related method of lipid mediated gene transfer is fusion with the bacterial protoplast. It's just like a shortcut. So what we do here is like we will insert our DNA, transfer the DNA into the bacteria. So transformation is back to the bacteria is easy than the eukaryotes, because eukaryotes are very sensitive. So whatever DNA we want to insert into the animal cell, we will transform it into the bacteria. Then we will fuse the bacteria with the animal cell, just like the cell fusion. Different types of bacteria as well as the cell will be fused. So after the fusion, the whole genome of the DNA, bacteria as well as our gene of interest will be in the cell. Inside the cell, the genomic DNA will be cleaved by the cellular mechanism. As we are inserting specific sequence in the DNA of interest, it won't be cleaved. So that's how we do the bacterial transformation, the transformation by a bacterial protoplast fusion. So we are fusing the bacterial protoplast with the cell. So that's all about the chemical methods of gene transfer. Now we have the physical methods of gene transfer for animal cells. So it include electroporation, ultrasound transfection, particle bombardment, laser-induced DNA delivery, or microinjection. Electroporation, the mechanism, setup, and everything is similar to the bacteria, but here we are using gentle techniques. We are using the electric current for a very brief time, and voltage gradient can be between 4000 to 8000 volt, but the period of delivery is really small. It induces the transient pore just like in the bacteria, and we can actually insert the DNA inside the cell. We use the pre-treatment to increase the transformation frequency. In eukaryotes, we can actually see the holes in the plasma membrane during the electroporation. Through these pores, we are actually inserting the DNA. And for ensuring the transformation, cells used to be arrested in the metaphase, because in the metaphase there is no cell nuclear membrane. Then only we can insert the DNA into the nucleus. So if it in a interphase, it will have the intact nucleus. Inserting DNA into the cell is difficult, but in the metaphase they don't have a nucleus, no nuclear membrane, so it is easy. And the transformation efficiency for the linear DNA is high in case of electroporation, but the input cost is high because the equipment we are using for electroporation is expensive for eukaryotes and especially for animals. But we also need large number of cells. The death rate of cells in the electroporation is high because the animal cells are very much sensitive. When we are introducing the electricity, cells will die. So we need large number of viable cells to get a small proportion of live cells after the method. One beauty of this method can be also performed in vivo. We can actually insert two electrodes into the skin, under the skin, and inject the DNA in between. So we can actually, this DNA after transferring a very high voltage electricity for a very brief time, like microseconds, the DNA will be get inserted into the human cells. But inside in vivo technique, these cells are more resistant than the in vitro condition. So the efficiency is high in case of in vivo technique, but in vitro, this method need huge number of cells because of the high toxicity. Next is ultrasound transfection. So we are actually exploring different ways in which we can put holes in this plasma membrane. Ultrasound is also able to make holes in the membrane. So we'll be subjecting the cells into very high frequency sound or very low frequency sound. Then it leads to the formation of bubbles in the lipid medium, which leads to the holes in the lipid medium, which will actually mediate the transfer of DNA from outside cell into the inside cell. So we can use comparatively low frequency ultrasounds in the waves both in vivo and in vitro. Just like we are inserting the electrodes, we can actually insert the probes for ultrasound into the skin or into the tissue, and we can inject the DNA in between and facilitate the transfer. That is particle bombardment. It is the same as the gene gun. Uh, it is a physical method, and it is used for the transfecting multiple cells in a tissue. It can be used in vivo or in vitro. It can also used for transforming mitochondria, the putting DNA into mitochondria. Next is laser-induced DNA delivery. In animals, we can actually use the laser-induced DNA delivery in three different ways. Either we can use high-intensity laser waves, which actually cause the holes, produce the holes in the membrane. Or we can use photochemical internalization. We can insert some type of photochemical sensitive dyes in the liposomes, and once it is inside a cell, we can actually excite it with using the light. So finally, we have the strong pressure wave. Pressure waves are actually a type of wave called photomechanical waves. It can the laser wave which can actually do the mechanical work. It's technically called laser induced stress wave. So laser induced stress waves can actually put holes in the cell membrane and mediate the DNA transfer. This is the setup of using leaves. So we have the mechanism for concentrating the laser, and we have the cell tissue. So once the wave is coming, has come, it will actually create holes in the plasma membrane of the membrane. So for laser-induced DNA uptake, we are using something called photosensitase. The photosensitase are activated by light. This one can be used for in vivo gene therapy. Like suppose we are making a lysosome, not liposome with the DNA, but we want the DNA to be transferred to a specific type of cell inside the human body, maybe the liver cell. What we will do, we will make the liposome using some photosensitizer. So there it has the lipids as well as the photosensitizer, and DNA will be inside that liposome, and it will be injected into the blood, and it will be go everywhere. But we want the transformation to occur only in the liver. When the liposomes are going all type of cells, they will take up the liposome and it will be in the endosome. So eventually this endosome will be mature into lysosome and cleave the DNA. But if I want to deliver the DNA in the liver, I will be exposing the liver to the particular wavelength of light. So when the light comes, the photosensitizer get activated and it will break open the lysosome, then deliver the DNA into the cytoplasm. And we can actually specify or target a drug or a DNA into the cell. So next method is actually unique for eukaryotes and especially for animal cells. It is called a microinjection. In the microinjection, we are using glass pipettes, very fine glass pipette, to physically inject the DNA into the cell. So in this, just like we are taking the injection, we are actually injecting a cell. A cell is very small, but we can manage to inject that. So here is a representation. We are using two micro manipulators. One is the holder and one is a needle. By using the holder, we are actually fixing the cell, and by using the needle, we are injecting the DNA solution into the cell. We can actually inject in the nucleus. So the this whole equipment is under the microscope, and in the microscope we have a Golden pipettes and we have a injecting needle. So we will be using the vacuum pump to actually hold the cell, and we will be injecting using the mechanical power, not with the hand but using the mechanical power, mechanical motors. We will be injecting the DNA into the cell. There are two different types of micro injection. One is constant flow, the flow rate is constant. Other is pulse flow. The pulse flow, the flow rate, the injection speed can be manipulated. It is used in pronuclear injection, which is used for making the transgenic animals. In the pronuclear injection, what we do, this is an egg cell immediately after fertilization. The sperm has came and fertilized. So inside the fertilized egg, in the initial stage, we have two different nucleus. This nucleus, the male nucleus, is actually called the pronuclei. So we will be collecting this type of cells and injecting our DNA into the pronuclei. And after the injection, the natural course, these two nucleus, the male and female nucleus, will fuse and producing the zygote, and we will be this transferring the zygote into a surrogate mother, and inside the mother this zygote will grow and it will eventually grow into a transgenic mouse. Whatever DNA you have injected into the pronucleus will be in this transgenic mice. It is a very simple mechanical process. Delivery is precise. We can see how we are delivering. Large amount of DNA can be delivered. That means there is no size restriction. It's a direct physical approach. We can be 100% sure that we have done it. Host independent, we can use it for any type of animal. The thing is that the cells should be larger, and the cytotoxicity is less comparatively, and regeneration capacity is high even after the injection. It is used for the production of transgenic animals, and the efficiency of transfection is very high. Finally, we have the biological methods or transfection mediated gene transfer. One of the classical examples for it is retrovirus mediated gene transfer. As we know that retroviruses are the viruses which is having the RNA genome but inside the cell it will be converting its RNA into the DNA using the reverse transcriptase. And we know about HIV virus, which is an retrovirus, which have the RNA genome, but it has the ability to convert the RNA into the DNA. So one of the peculiarity of retrovirus is like its DNA used to be integrated into the host genome. Actually, its genome is RNA, but inside the cell, it will convert the RNA into the DNA, and this DNA has a tendency to get inserted into the genomic DNA, which can be exploited for making the virus vector. We can use the recombinant retroviral vectors for transformation by using methods. It is like we are making a infectious transformation, so it can be used for transfecting many cells simultaneously, or we can use it for a in an organism, in a living organism. But sometimes it may use chimeras because some area of the organism will be affected, but the other areas won't be affected. Technically, this method is simple, but making this type of virus is actually difficult. It's actually like making a bioweapon. So we use the recombinant viruses for viruses which is genetically engineered, which can actually carry therapeutic genes or other type of sequences which will be delivered into specific cells. And it is extensively used though and researched in human gene therapy. And the most commonly used retrovirus is not HIV virus. We cannot use the HIV virus here, but we are using Molony-Murine leukemia retrovirus for these experiments. But the thing is that recombinant retrovirus cannot replicate by itself, so its infectious possibility is less. It is actually disarmed. So the retrovirus is actually a two-part system, just like the other viral system. It has a retroviral vector and a packaging cell line. The retroviral vector can infect any type of cell. It's like a wild type derived from the wild type, but it has a genome inserted into the uh, foreign DNA is inserted into the genome. So it has almost all type of sequence, but the uh, it cannot replicate in all type of cells, but it can replicate only in packaging cell line. So when the viruses is infected in a packaging cell line, it will produce viable viruses. But this virus can actually infect any type of cells, but it do not replicate. Okay, so the genome of the virus containing our gene of interest, not the viral genome required for the replication of DNA or the production of the viral particles. The production of viral particles is done in the laboratory or in the industry using packaging cell line. So from the packaging cell line, we will get viable viruses. These viruses can be used for gene therapy. So this virus will actually have the RNA genome. It will come inside and convert this RNA into the DNA, and this DNA can be inserted into the genome. So this is the same thing. Even though this retroviral system is very efficient and effective, there are some limitations, which include transfection efficiency is low, then unable to infect non-dividing cells, affect only dividing cells. The carrying capacity is 8 kb, and targeting is difficult. It is actually to infect any type of cells. So there are methods for transgecting, but it is difficult. So these are the methods we are used for transformation in animal cells. In this session, I will be talking about transformation to plant cells. Thank you very much.